Hey y'all, I'm Cal. Let's get messy. Hi guys, welcome back. Today we are going to talk about something that is super exciting, and that is the new research report that was just released by Princeton University on July 9th in Nature Genetics, and what it means. I spent about 2 days going through it, trying to figure out what all of the different parts mean and breaking it down so that I could understand it. And I wanted to share it with you. So, because this research, I believe, has the power to change how we not only understand, but diagnose and support autistic people. So I think it's very important that we understand exactly what this report means. I'm going to share with you my understanding of it. And I would love to hear in the comments if I got anything wrong, or you have a different viewpoint on it, or you have additional research or information that you could share with everyone. But I do want to be clear before we jump into this. The study is not suggesting that autistic people need to be fixed or that there's anything wrong with being autistic. From my viewpoint, what it's doing is giving a more nuanced understanding of autism's biological diversity. And again, that could lead to better support, earlier identification of needs, more personalized approaches to care. I've got some infographics that I'll pop up somewhere in here, and I'll also put a link to them down in the description, as well as a link to the study findings so you can read it for yourself. I highly suggest you read it and do your research, figure out what you think it means, and please share it. So, the research, what did they actually do? The study was led by researchers at Princeton University and the Simons Foundation, and it is massive in scope. Data from over 5,000 children from the SPARK study, which is one of the largest autism research cohorts in the world. And what makes it different from previous research is that instead of looking at single traits or genes in isolation, they used a person-centered approach. Instead of studying like individual pieces or characteristics and trying to fit them into each person, they're looking at a bunch of characteristics from each individual person, all of those being the same. I think there were 239 different traits for each child, and it was everything from social interactions, repetitive behaviors, developmental milestones, co-occurring conditions, and then they used advanced computational modeling to find patterns in how these traits cluster together. So, for example, you'll have this chunk of traits over here and this chunk of traits over here, and how did these chunk of traits connect with these chunk of traits, and they put it all together and they found a pattern. And they broke it down into 4 distinct subtypes, each with its own profile of traits, genetic patterns, developmental trajectories. And, we're familiar with subtypes. They've been looking for that for years, but we'll get into that later. But the 4 subtypes, what are they? The first subtype is social and behavioral challenges. This subtype was the largest group out of all the participants. It was 37%. This group had core autism traits like social difficulties and repetitive behaviors. They often had co-occurring ADHD and anxiety. The key to this group was that they reached developmental milestones at typical rates, which meant also that they were usually diagnosed later than other groups. And this group had genetic mutations in genes that became active later in childhood, not before birth, like we've thought up until now. Second type is mixed ASD, mixed autism spectrum disorder with developmental delay. And this only affected 19% of the participants. So what this was delays in reaching milestones like walking and talking. Mixed presentation of repetitive behaviors and social challenges. What I found interesting about this group is that they generally don't show high levels of anxiety or oppositional behavior. This group also had both inherited genetic variants, and new mutations. Subtype 3, moderate challenges. This is the next largest group of participants at 34%. They had lower scores across all measured categories compared to other autistic groups, but they still were significantly different from their non-autistic siblings. That's another thing about this study was they not only studied those who had autism, but also their siblings. But this group also had mutations in genes that are less evolutionarily constrained. When scientists talk about evolutionarily constrained genes, they're talking about genes that are so critical for survival that they rarely change across evolution, right? So think of highly constrained genes like your critical system files on your computer, the files that you need to run. If you mess with them, everything breaks down. The moderate challenges group had mutations in genes that are less constrained, meaning these were genes more like our preference settings rather than critical system files. So these are genes that can tolerate changes without causing major developmental disorders. This might explain why this group has moderate rather than severe challenges. Their genetic changes are affecting less essential systems, allowing for better overall functioning, while still causing meaningful differences in brain development. Subtype 4. These are called the broadly affected, and this was the smallest group of participants at about 10%. And this group had high difficulties across all areas measured. They had the highest rates of intellectual disability and developmental delays. They were most likely to have co-occurring conditions. They had the highest burden of new genetic mutations, particularly in the highly constrained genes that we just talked about. So think of them as what we would now call level 3, high support needs. So what does all of this tell us about biology? So each subtype had distinct genetic signatures, different patterns of mutations and variations. And the research found that the broadly affected group had the most new genetic mutations, de novo mutations, meaning they're not inherited from parents, but occur spontaneously. And these mutations were specifically in genes that are crucial for brain development. And the bombshell finding in this whole study was the timing of when the genes are active during development corresponds to when symptoms appear clinically. So they used to think that most autism-related genetic effects happened before birth, right? But the social and behavioral challenge group, the ones diagnosed later with fewer developmental delays, had mutations in genes that become active later in childhood, not at birth. And it's really important that we understand this part correctly because we're talking about when genes become active, right? Not when genetic mutations occur, because I can see the anti-vaxxers just going wild over this point without fully understanding what it means. The genetic variations are still present from birth. They just affect genes that turn on at different developmental stages. So it's like having a genetic timer was set from conception, but some effects don't show up until later when those specific genes start doing their job in brain development, right? This suggests that for some autistic people, biological mechanisms of autism might emerge after birth, which aligns perfectly with their later diagnosis and different developmental patterns. And this is important because if we understand when different genetic programs are active, we can develop more targeted and timely interventions. Researchers have been trying to identify meaningful subtypes for decades, but previous studies have had significant limitations. Wing and Gould's 1979 study identified 3 social subtypes: aloof, passive, and but odd. Studies have identified subtypes based on cognitive abilities, sensory features, severity of core symptoms. And most studies were much smaller and used much simpler analytical approaches. So why is this study different? In this study, they used over 5,000 participants versus, hundreds in most previous studies. This study was person-centered rather than trait-centered. This is the first study to link autism subtypes to specific genetic programs. Another thing that was really fascinating is they replicated their findings 100% in an independent cohort. They connected genetic activity timing to clinical presentations. Previous studies, like I said, were more trait-centered, looking at individual characteristics rather than whole person profiles. They used limited genetic data. They had smaller sample sizes. And quite honestly, they had a lot of difficulty replicating results across different populations. So how was this study validated? Well, they did it through the Simon's Simplex collection, which is a completely independent group of families. And when your findings are replicated in a completely different population, that's when you know you're onto something. So what could this mean? We don't want to get too ahead of ourselves. I'm talking like 10 years out, not tomorrow. We're not going to see the effects of this immediately, but there's so much potential there that we haven't seen before. More precise, personalized diagnostic approaches identify children who need support earlier. This could reduce the current average diagnostic delays that we see. For those of you who have been diagnosed or gone through the diagnosis process, you know how long it is, especially for girls and people of color. It could affect support and intervention. So each subtype might benefit from different types of support. Social and behavioral challenges group might need more focus on anxiety and ADHD management. The developmental delays group might need more intensive early intervention. It can move us away from what we have now as this sort of one-size-fits-all approach. Like, if you're autistic, do this. If you're autistic, you need this. That's not working. Families might have better information about what to expect. It could help with educational planning and life preparation, reduce uncertainty and anxiety for families. Drug companies could develop treatments targeted to specific subtypes, and I'm speaking specifically of ADHD and anxiety-related medicine, not medicine to cure autism. Make that clear. Research studies could be more precisely designed, could accelerate discovery of new intervention. What it doesn't mean is that autistic people need to be cured or fixed. It doesn't create a hierarchy of autism subtypes, which is what I feel like we have now. A lot of people in the community feel that. It doesn't reduce the importance of neurodiversity perspectives. It doesn't mean we should start labeling and segregating autistic people. What it does mean is that, autism is biologically diverse. Personal support may be more effective. Individualized differences have biological basis. Understanding can improve outcomes. And genetics explains autism diversity. Getting into that, some people might wonder, like I did when I first read it, if genetic effects emerge after birth, does this give any validity to claims that vaccines or other environmental factors cause autism? Absolutely not. I think we need to understand why this distinction is crucial. It found that genetic programs become active at different times, but genetic variations themselves present from birth or conception. So it's like, having all of the ingredients from a recipe to start, but some steps in the cooking process happen later, right? The ingredients don't change just when they get used. And so the genetic variants identified in the study are inherited or occur during very early development. And we know that vaccines cannot alter your DNA or create these genetic patterns. And what this research actually shows is that it strengthens the evidence that autism is primarily genetic by showing us exactly how genetic programs unfold during development when genetic influences become apparent, not what causes autism. I believe this should increase our confidence in genetic explanations for autism, not decrease confidence in vaccine safety. I just really think it's important to be clear about this because misunderstanding genetic timing research can potentially harm public health, and that's not what this science is showing us at all. But I do think we need to talk about its limitations because no study is perfect. So, the data. Most information came from parent reports, so it was self-reported, which can have biases. Population diversity. While the study did include diverse families, autism research has historically underrepresented certain communities. Another thing is this is a snapshot in time, right? So they're not tracking changes over development. They're looking at specific snapshots in time, kind of like a balance sheet versus a P&L statement, if you're familiar with accounting. It was also focused on children. We don't know if these subtypes remain stable into adulthood. Do they change as people age? How do environmental factors interact with these genetic subtypes? Will these findings apply across cultural context? How might these subtypes relate to autistic people's own experiences and self-understanding? While they did validate in one independent cohort, it needs to be replicated across multiple populations, right? International replication will be crucial to studies following people over time. The researchers themselves acknowledge these limitations, which is, to me, a really good sign. It shows that they're being scientifically responsible. I think we're at a crucial point where we need to include perspectives from the autistic community on this research. While I haven't seen a bunch out there yet because, again, this just came out less than a week ago. But, based on previous similar research, there's typically mixed reaction. Increased medicalization of autism, concern about subtyping being used to create hierarchies, fear that this could reduce support for some autistic people, particularly with those, in the social and behavioral challenges or the moderate challenges categories. Questions about whether this research reflects autistic people's priorities, right? But there are benefits too. It can lead to more appropriate support. It gets us talking. It could reduce misunderstandings about autism's diversity. Again, earlier identification and intervention is so important. I think it might help validate experiences of autistic people who don't fit the typical descriptions, right? I don't feel like I fit a lot of the typical descriptions, and that's a struggle for me that we'll talk about on another day. But what the autism community has historically valued, is research that includes autistic researchers and perspectives, studies that focus on improving quality of life rather than normalizing it, recognition of autism as a neurological difference, not a disease to cure, emphasis on accommodations and acceptance rather than just intervention. It's worth noting that this research was conducted with significant community input and oversight through the SPARK Consortium, which includes autistic self-advocates. So now that we have kind of a basic understanding of what this report means, where do we go? Well, the researchers laid out several important next steps. Really, it feels like a research revolution rather than an end. Longitudinal studies following these subtypes over time to see if they remain stable. Intervention studies, testing whether subtype-specific approaches work better. Adult populations, extend this research into autistic adults. Diverse populations, again, ensuring that these findings apply across different ethnic and cultural groups internationally. But there are some technological advances needed first. Development of biomarkers that could aid in subtype identification, digital tools for tracking development and outcome, assessment tools for subtype identification, training for clinician, guidelines for subtype-specific interventions, ensuring that subtypes aren't used to discriminate or limit services, maintaining focus on people's own goals and preferences, and balancing the medical model's benefits with neurodiversity perspectives, protecting against genetic discrimination. The researchers estimate that we could see clinical applications within the next 5 to 10 years. Historically speaking, that seems optimistic to me. Real world implementation of research like this typically takes much, much longer. I don't believe I'll see any of this in my lifetime, but that's not my concern. But this study does feel different. The scale, the methodology, the integration of genetics and behavior. I feel like it's answering request that we've had for a long time. But I also know that genetic research feels threatening, especially in this political climate. Because no matter what your political leanings, whether you want to admit it or not, we have a history of genetic research being used to justify harmful practices. You can't deny that. And so that is a fear. What gives me hope about this research is that it explicitly frames this as an understanding of biological diversity, not deficits. The focus is on improving support and outcomes, not elimination or cure. The methodology respects the complexity and individuality of autistic people in general. We're just people. It was conducted with community oversight and input. So that gives me hope. More concerns, how will the information be used by insurance companies, schools, service providers? Will one subtype be seen as more valuable or more deserving of support? How do we ensure this research benefits all autistic people, not just those who fit into, neat little categories? I think the key is that this research should enhance our understanding of autism as a neurological difference that brings both challenges and strengths. So what does this mean for you, me, us? It validates what we've known for a long time. Autism is incredibly diverse. Our individual pattern of traits is meaningful and has a biological basis. It doesn't change our value or our need for support. And future research might lead to better, more personalized support options. So that's my take on it, guys. I hope you will leave some comments. Let me know what you found. Let me know where you disagree with me. Teach me something, please, because this is important for all of us to understand and to learn and to share. So I hope you'll share it. And let's keep talking about it. And if you would like to be on the podcast and discuss this, there is a link in the description below. And I hope you have a great week. I don't have anything else to say. Love you. Bye.